Hi, my name is Yoonjung Kim, and I'm going to present the genomic insight into polymycoviruses in-depth analysis of diversity and evolution in the Polymycoviridae family. First, we gathered the latest data from the NCBI virus dataset, which you can find at the website. Then we cleaned the data, removing any missing value and outliers to ensure smooth analysis and visualization. This gave us a master data set ready for analysis. We used Excel and ProtParam as our main tool. In total, we collect data for 307 polymycovirus nucleotides and 56 RDRP proteins. When we look at the data, we found that China submitted the most polymycoviruses sequences, which you can see in figure 1. The most frequently occurring virus was Bostreopharia polymycovirus 1 with 23 cases, followed closely by Cetosperia turcica polymycovirus 2 with 20 cases. For reference, Bostreopharia and Axillahilum tursicum, the host fungi for these viruses, both belong to the Ascomycota phylum. Across all data, regardless of country, Cladosporium cladosporioides species was the most infected with polymycoviruses, which you can see in figure 2. Then we investigated the physicochemical properties of the RDRP protein of polymycoviruses in table 1 to uncover hidden correlationship between these variables. We performed exploratory factor analysis clustering similar variables. We used SPSS for this analysis. We applied Butler's test of sphericity, principal component analysis, and varimix rotation. Our analysis showed that Butler's test was significant, indicating that our factors were well defined. Typically, a communality value above 0.4 conforms extraction, and in our study, all above exceeded this threshold. The explanatory power of our model is illustrated in figure 3. The rotation component matrix from PCA grouped the parameters into 3 categories. Group 1 is length, molecular weight, and negative positive charge, atomic composition of carbon, hydrogen, nitrogen, oxygen. Group 2, atomic composition of sulfur. And group 3, AI gravity. We excluded the theoretical PI from this grouping. Since group 1 had high explanatory power, we used discriminant analysis to compare this parameter with other dsRNA viruses to see if they could help detected polymycoviruses in table 4. We found that the 8 selected physical and chemical characteristics were statistically significant for identifying polymycoviruses. Wilk lambda test lambda was 0.109 and chi-square was 126.087, showing significant discriminant score between groups. The canonical correlation coefficient indicating the relationship between the discriminant variable and the actual group was 0.944. Molecular weight was the most important variable for identifying polymycoviruses, while the atomic composition of carbon and oxygen were less relevant. The classification accuracy or hit rate for distinguishing between polymycoviruses and other dsRNA viruses was 100%, demonstrating high accuracy. As a result of the analysis, we found that among these physicochemical properties of polymycoviruses, length, molecular weight, negative, positive charge, and atomic composition of hydrogen and nitrogen were relatively important. Thank you.